Welcome to the deep dive. Imagine you're just going about your day and suddenly you need a burst of energy. Maybe you're sprinting for a bus or, your brain needs to fire on all cylinders to crack a tough problem.
Yeah, where does that immediate fuel actually come from?
Exactly. It's not your last meal, not right that second.
Okay.
And it's not from your vast fat reserves either. They're more for the long haul.
That immediate on-demand energy comes from a really remarkable molecule. Your body's incredibly efficient, ready-to-use fuel reservoir, glycogen.
And that's our mission today. We're taking a deep dive into Chapter 15 of Lehninger's Principles of Biochemistry, the metabolism of glycogen in animals. And this isn't just about, simple sugar storage. It's a fascinating look at one of the most critical energy management systems in vertebrate biology.
Our goal really is to unpack the remarkable molecular mechanisms and the exquisitely coordinated regulatory pathways that govern glycogen. We'll explore its unique structure, how it's swiftly built and broken down, and the intricate dance of hormones and enzymes that keeps your energy levels, well, perfectly balanced.
Here's a key insight right from the start, which I think is fascinating. While your body stores about 100 times more energy as fat than as glycogen.
Which is a huge difference.
Huge. But fats cannot be converted to glucose in vertebrates. And crucially, they also can't be used for quick anaerobic energy bursts.
Exactly. The kind your muscles often need during really intense activity.
So that's where glycogen comes in.
That's why glycogen is unique. It provides immediate, fast-access glucose. Crucial for those quick bursts of activity, especially for your brain, which, as you said, relies almost entirely on glucose.
Right, can't use fats.
And your muscles, which need that rapid fuel for contraction. Think of it as your body's sprint fuel, maybe, not its marathon reserve.
Okay, so if glucose is so vital, why doesn't your body just store it as individual glucose molecules? It seems simpler, right?
It does seem simpler on the surface, but there's a critical problem with that approach. Osmolarity.
Ah, the water problem.
Precisely. If a cell tried to store enough free glucose to equal the energy density of stored glycogen, the internal concentration of sugar would be incredibly high.
So water would just rush in?
Water would rush into the cell. Following the concentration gradient, it would swell and, quite literally rupture. It's just not a viable storage strategy for the cell.
So the body's elegant solution is to polymerize glucose into glycogen. Tell us about this branched solution.
Well, glycogen is stored as these compact granules inside your cells, mostly in your liver and muscles. And its structure is incredibly ingenious.
How so?
It's a polymer made of glucose units linked together, but with branches sprouting off the main chains. These branches create a very dense, almost tree-like structure.
And that branch design is more than just about packing it in tightly, isn't it?
Absolutely. A typical glycogen granule can hold, tens of thousands of glucose units. But here's the really crucial part. Its branched design creates thousands of non-reducing ends.
Non-reducing ends, what does that mean in practical terms?
Think of these as thousands of accessible active sites or working points all over the surface of the granule.
Ah, okay, so lots of places for enzymes to latch on.
Exactly. This vast number of accessible ends allows multiple enzymes to work simultaneously, either breaking down or building up the glycogen. This means incredibly rapid glucose release when you need it.
Faster than if it were just one long chain.
Much, much faster, and all without the osmotic issues of storing free glucose. It's a brilliant design.
And we see very specific roles for glycogen, depending on where it's stored. You mentioned liver and muscle, a real tale of two organs. Let's start with muscle.
Okay. Your skeletal muscle holds a significant amount of glycogen, maybe up to 400 grams in an adult.
That's quite a bit.
It is. And the key thing here is that muscle uses its glycogen exclusively for its own energy needs.
So it's selfish glycogen.
In a way, yes. It provides immediate fuel for those sudden bursts of activity, like when you're lifting something heavy or need to move quickly. The glycogen there is really designed for rapid, localized energy release.
And the liver, how does its role differ?
The liver's role is quite different. It stores less overall, maybe about 100 grams, but that accounts for a higher percentage of its actual weight.
Okay.
And the liver's primary role is to act as the body's central glucose bank, you could say. It maintains stable blood glucose levels for the entire body.
Especially for the brain.
Especially for your brain, absolutely. Unlike muscle, the brain cannot use fats for fuel and depends on a constant glucose supply. So liver glycogen release is typically slower, more steady, fitting its role of constantly replenishing blood glucose between meals.
And right at the core of all this, sort of kicking the whole thing off, is a fascinating protein called glycogen.
Yes, glycogen is really unique. It's found at the very center of every single glycogen granule.
What does it do?
It acts as both a primer and an enzyme. It kickstarts the assembly of new glycogen chains. It actually adds the first few glucose residues to itself onto one of its own amino acids, forming a short starter chain. Then the main glycogen-building enzyme can take over and extend it. It's the essential foundation, the seed, for every new glycogen molecule.
Wow. Okay, so now that we've seen how glycogen is structured and stored, let's look at how it's broken down. The process called glycogenolysis.
Right.
And there's an interesting distinction here, isn't there, between how your body breaks down its stored glycogen versus, say, the starch you eat.
That's a great point. Dietary carbohydrates, like starch, are broken down by enzymes in your gut, mainly through hydrolysis. That means using water to break the bonds.
Okay.
But the glycogen stored inside your cells is degraded by a different and actually quite clever mechanism called phosphorylasis.
Phosphorylasis. Using phosphate. What's the clever part about that?
Well, with phosphorylasis, the main enzyme, glycogen phosphorylase, uses inorganic phosphate pi, just floating around in the cell, not water, to cleave the glucose units off the chain.
And it releases them as
It releases them as glucose 1-phosphate. And here's the metabolic genius, the really neat trick. By using phosphate instead of water, the body essentially preserves some of the energy from that broken bond right there in the glucose 1-phosphate molecule.
so it's like pre-activating it for the next step.
Exactly. It's like getting a prepaid ticket for the metabolic highway.
Yeah.
This pre-activated glucose 1-phosphate is instantly ready for further energy extraction, and crucially, it saves the cell an ATP molecule it would otherwise have to spend later on.
Very efficient. A metabolic shortcut.
It's a remarkably efficient shortcut, yes.
So glycogen phosphorylase works its way along the glycogen chain, snipping off glucose 1-phosphates.
Yeah.
But it doesn't do the job alone, right? Especially when it hits those branch points we talked about.
Correct. Glycogen phosphorylase chews along the straight linked chains, but it stops its action. Usually about 4 glucose residues away from a Pence branch point, it gets stuck there.
So you need help.
You need the debranching crew, essentially.
Yeah.
A bifunctional debranching enzyme steps in. It has 2 distinct activities.
Okay, what does it do?
First, it acts like a transferase. It shifts a little block of 3 glucose residues from the branch over to the end of a nearby chain, reattaching them with a normal link.
Right, extending the main chain.
Exactly. Then, its second activity comes into play. It hydrolyzes, this time using water, the single remaining glucose unit that was attached at the original IR16 branch point. This releases it as free glucose, not glucose 1-phosphate.
Interesting. So you get mostly glucose 1-phosphate, but a little bit of free glucose too.
A small amount, yes. But the main product is glucose 1-phosphate, and removing that branch allows glycogen phosphorylase to get back to work on the now longer unbranched chain.
Okay. So once we have this pool of glucose 1-phosphate, it needs a little modification, and then its journey really diverges depending on which tissue it's in, muscle or liver.
Precisely. Glucose 1-phosphate is first quickly converted to glucose 6-phosphate by another enzyme, phosphoglucomutase. This is easily reversible.
Glucose 6-phosphate. That's a familiar molecule from glycolysis.
It is. And now, the path for glucose 6-phosphate truly splits based on the cell's needs and location.
In muscle, it seems pretty straightforward, straight to energy, right?
Yes, in muscle, the goal is immediate energy for contraction. So glucose 6-phosphate directly enters the glycolysis pathway.
And it skips the first step of glycolysis.
It does. It enters partway down, bypassing the initial step that usually requires ATP investment. This makes glycogen breakdown an even more rapid and slightly more energy-efficient source for muscle activity.
Makes sense. But in the liver, the purpose is entirely different.
Absolutely. In the liver, the primary goal isn't to use the glucose for its own energy. It's to release free glucose into the bloodstream to maintain those blood glucose levels for the rest of the body, especially the brain.
So how does it get the phosphate off the glucose 6-phosphate?
This requires a specific enzyme called glucose 6-phosphatase. And here's a key point. This enzyme is found almost exclusively in the liver and kidneys, not in muscle or brain.
Tissue specificity.
Exactly. And there's another neat trick with its location within the liver cell.
Oh, where is it?
It's located inside the endoplasmic reticulum, or ER, which is a network of membranes within the cell. Glucose 6-phosphate has to be specifically transported into the ER lumen.
Why put it in there?
It's compartmentalization. Once inside the ER, glucose 6-phosphatase clips off the phosphate, producing free glucose. This free glucose is then transported out of the ER and subsequently out of the liver cell into the bloodstream via specific transporters like GLUT2.
That's clever. What's the advantage?
It's brilliant because it keeps the newly generated glucose 6-phosphate physically separate from the glycolytic enzymes in the cytosol. This ensures it doesn't just get immediately used up for energy within the liver itself, it maximizes glucose export.
So it guarantees the glucose gets out to where it's needed.
Precisely. And just as a side note, genetic defects in this glucose 6-phosphatase enzyme or its transporters lead to serious metabolic conditions, the glycogen storage diseases, because the liver can't release glucose properly.
It really highlights how critical this pathway is. And it's incredible how much we know about these intricate processes. We owe a huge debt to some pioneering scientists, don't we?
We absolutely do. Much of our foundational understanding of glycogen metabolism, especially the breakdown pathways, came from the incredible work of a husband and wife team, Carl and Gertie Corey.
The chorus. I've heard their name.
Between roughly 1925 and 1950, they just meticulously uncovered these pathways. They discovered key enzymes like glycogen phosphorylase, identified critical intermediates like glucose 1-phosphate, sometimes called the Corey ester.
Wow.
Their work was truly groundbreaking and even inspired other major discoveries like Arthur Kornberg's work on DNA polymerase and Gertie Corey's later work on identifying the biochemical defects in human genetic glycogen storage diseases was also monumental.
They share the Nobel Prize in 1947, right? A real legacy.
A true legacy, yes. Their work laid so much groundwork.
Okay, moving on to the other side of the coin then. Building glycogen back up. Glycogen synthesis or glycogenesis. This process also involves some clever molecular strategies, starting with what's called an activated donor.
Yes, exactly. Instead of just trying to stick glucose molecules together directly, which is energetically difficult, the body uses an activated form of glucose called UDP glucose.
UDP glucose. What's the UDP part?
It stands for uridine diphosphate. It's a nucleotide, similar to ADP or GDP. So it's basically glucose attached to UDP. This sugar nucleotide is beautifully suited for building complex carbohydrates like glycogen for several key reasons.
Okay, what makes it so special?
Well, first, its formation is metabolically irreversible. When UDP glucose is made, another molecule called pyrophosphate, or PPI, is released.
Right.
This pyrophosphate is immediately broken down by another enzyme, inorganic pyrophosphatase. And that breakdown releases a lot of energy. It's strongly exergonic.
So that pulls the first reaction forward.
Exactly. It pulls the overall synthesis reaction strongly in the direction of making UDP glucose. This strategy of rapidly removing a product to ensure irreversibility is a really common and elegant trick in biological polymerization reactions. It's like slamming a one-way door shut.
A one-way street, ensuring the reaction keeps going towards synthesis.
Precisely. Secondly, that attached UDP nucleotide acts like a handle. It provides multiple points for enzymes to grab onto through non-covalent interactions, which contributes to binding energy and catalytic specificity.
Okay, like a tag?
Sort of. And third, the UDP moiety itself is an excellent leaving group chemically. This means it activates the glucose molecule, making it much easier, energetically speaking, to add that glucose onto a growing glycogen chain.
Easier to transfer the glucose.
Much easier. And finally, you could say that by tagging glucose with UDP, cells can specifically earmark it for glycogen synthesis, keeping it separate from the glucose destined for immediate energy use via glycolysis.
So it directs traffic.
It helps direct the metabolic traffic, yes.
Okay, so how does this step-by-step construction of glycogen actually work using this UDP glucose?
It generally starts with glucose 6-phosphate. This can come from glucose entering the cell from the diet or in the liver. It can even be made from precursors like lactate via gluconeogenesis, like in the Corey cycle.
Right, linking different pathways.
Exactly. This glucose 6-phosphate is first converted back to glucose 1-phosphate by that phosphoglycoamidase enzyme we mentioned earlier.
Okay.
Then comes the crucial activation step. An enzyme called UDP glucose pyrophosphorylase catalyzes the reaction between glucose 1-phosphate and UTP, another nucleotide triphosphate like ATP, to form UDP glucose and that pyrophosphate, PPI.
And the PPI gets destroyed, pulling the reaction forward.
Instantly hydrolyzed, yes, making the formation of UDP glucose essentially irreversible in the cell.
Okay, now we have our activated glucose. What next?
Now the main player, glycogen synthase, takes over. This is the enzyme that actually elongates the glycogen chain.
How does it work?
It transfers the glucose residue from UDP glucose onto one of those non-reducing ends of a pre-existing glycogen chain, forming a new glycosidic linkage. UDP is released, and the glycogen chain gets 1 unit longer.
And this happens over and over.
Over and over again. The overall process, starting from glucose entering the cell all the way to adding it to glycogen, strongly favors building up those glycogen reserves when glucose is plentiful.
But glycogen synthase can't build those critical branch points we talked about earlier, right? That requires something else.
Correct. Glycogen synthase is like a worker who can only build straight roads. It's limited to making the disoyl linkages. The branches, the 6 linkages, are formed by a different specific enzyme called the glycogen branching enzyme.
That comes along and adds side streets.
That's a good analogy. It works by taking a terminal fragment, usually 6 or 7 glucose residues long, from the end of a growing chain, breaking a NIT1,4 bond, and transferring that whole block to an interior glucose residue on the same or another chain, attaching it via an A6 linkage.
Creating a new branch point.
Exactly, creating a new branch point.
And why is that branching so important? Again, beyond just compacting the structure.
It's absolutely crucial because, as we discussed before, it dramatically increases the number of non-reducing ends.
More ends for enzymes to work on.
Precisely. Both glycogen phosphorylase, for breakdown, and glycogen synthase, for synthesis, act only at these non-reducing ends. So, more branches mean exponentially more accessible sites for both processes. It hugely enhances the efficiency and speed of both breaking down glycogen when needed and building it back up when possible.
It makes the whole molecule much more dynamic.
Much more dynamic and responsive, yes.
And circling back to our friend glycogen at the core, it's not just sitting there after the granule is built. It's actually involved in the very first steps of synthesis, isn't it? You mentioned it was a primer.
Yes, that's right. Glycogen synthase, the main chain builder, cannot initiate a new chain from scratch. It needs something to add on to, a primer.
And glycogen provides that.
Glycogen is the primer, and it makes the primer. It's really unique. It's an enzyme that catalyzes the addition of the first few glucose residues, usually about 8 from UDP glucose onto one of its own tyrosine amino acid residues.
It builds onto itself.
It builds the initial short chain onto itself. Once this primer chain is long enough, glycogen synthase can then bind and take over, extending the chain much further. So glycogen is essential for initiating the synthesis of every single new glycogen molecule.
What a coordinated effort. Just incredible molecular machinery. And this leads us perfectly into the final section, the symphony of coordinated regulation. Glycogen metabolism must be tightly controlled.
Oh, absolutely. It's a prime example, a textbook case, really, of exquisite metabolic control. In fact, glycogen phosphorylase was one of the very first enzymes discovered to be regulated both allosterically.
Meaning molecules binding away from the active site.
Right. And also controlled by reversible phosphorylation, the addition or removal of phosphate groups. These layers of control, involving enzyme cascades, ensure that glucose is stored or mobilized precisely when and where it's needed without waste.
Okay, let's break that down.
Yeah.
Starting with the regulation of glycogen phosphorylase, the breakdown enzyme, how is its activity switched on and off?
Okay, the primary on switch comes from hormones. In muscle, it's primarily epinephrine, the fight or flight hormone. In the liver, it's mainly glucagon, the hormone signaling low blood sugar.
And how do these hormones flip the switch?
They trigger an enzyme cascade. It's a beautiful amplification system. A hormone binds to its receptor on the cell surface, triggering a signaling pathway inside the cell, usually involving cyclic AMP or KMP.
Right.
KMP activates another enzyme, protein kinase A, PKA. PKA then phosphorylates and activates another kinase called phosphorylase B kinase, and this kinase finally phosphorylates glycogen phosphorylase itself, converting it from a less active B form to a highly active A form.
Wow, a cascade. So a tiny initial signal gets amplified hugely.
Hugely amplified at each step. A very small amount of hormone binding can lead to the activation of a massive amount of glycogen phosphorylase, resulting in a rapid flood of glucose, 1-phosphate.
Makes sense for a quick energy response.
Yeah.
And are there other controls too, especially in muscle, that fine-tune this? Things happening right inside the cell?
Yes, definitely. In muscle, there are crucial allosteric controls. calcium ions, Ca2+, which are the direct signal for muscle contraction itself.
Right, released when the muscle is told to contract.
Exactly. Calcium directly binds to and helps activate that phosphorylase B kinase enzyme, even without the hormonal signal sometimes. It links muscle activity directly to fuel mobilization.
Clever.
Also, AMP, adenosine monophosphate, which accumulates during vigorous muscle activity when ATP is being used up rapidly, is a sign of low energy charge. AMP directly binds to and activates glycogen phosphorylase B boosting breakdown.
So when energy is low, turn on the fuel supply.
Precisely. And conversely, when ATP levels are high, indicating plenty of energy, ATP competes with AMP for that binding site and essentially turns phosphorylase off, preventing unnecessary glycogen breakdown.
Okay, and the liver's phosphorylase has its own unique trick, acting as a kind of glucose sensor itself.
Yes, this is fascinating. The liver's active phosphorylase A form has a binding site for glucose itself. When blood glucose levels rise and glucose enters the liver cells.
After a meal, for instance.
Exactly. Glucose binds directly to phosphorylase A. This binding causes a conformational change that makes the enzyme a much better target for a phosphatase enzyme, we'll talk about that next, which removes the activating phosphate group.
So high glucose essentially tells the liver, okay, stop breaking down glycogen now, we have enough.
Precisely. It inactivates phosphorylase, slowing glycogen breakdown and preventing the liver from releasing even more glucose into the blood when levels are already sufficient. It's a direct feedback inhibition.
And you mentioned a phosphatase. That's the off switch.
Yes. The main off switch for phosphorylase is an enzyme called phosphoprotein, phosphatase 1, or PP1. It removes that activating phosphate group from phosphorylase A, converting it back to the less active B form.
Okay, now let's flip to the other side. Glycogen synthase, the enzyme that builds glycogen. Its regulation must be coordinated with phosphorylase, right? Likely the opposite.
Largely the opposite, yes. It makes perfect sense. You don't want to be building and breaking down glycogen intensely at the same time.
Right, futile cycle.
Exactly. So, unlike phosphorylase, the active form of glycogen synthase, called synthesase, is the one that is unphosphorylated. Its activity is actually inhibited by phosphorylation.
So adding phosphate turns it off.
Correct. Several different protein kinases can phosphorylate glycogen synthase at multiple sites, leading to its inactivation, converting it to the B form. A key kinase involved here is glycogen synthase kinase 3, or GSK3. And during metabolic stress, AMP-activated protein kinase, AMPK, also inhibits synthesis.
So how does insulin fit into this? Insulin signals high blood glucose, so it should promote storage, right?
Exactly. Insulin plays a crucial role in activating glycogen synthesis. It does this mainly through a signaling pathway that leads to the inhibition of that inhibitory kinase, GSK3.
So it inhibits the inhibitor.
Yes, it takes the brakes off, effectively. Insulin signaling also leads to the activation of that phosphatase, PP1, which removes the inhibitory phosphate groups from glycogen synthase, directly activating it.
Okay, so insulin strongly promotes glycogen building. Does glycogen synthase also have its own allosteric sensor, like phosphorylase did with glucose?
It does, but it senses glucose 6-phosphate. When glucose 6-phosphate levels are high inside the cell, meaning plenty of glucose has entered and been phosphorylated, it acts as an allosteric activator for glycogen synthase B, the less active phosphorylated form.
What does it do?
It binds to synthase B and makes it, one, slightly more active itself, but perhaps more importantly, it makes it a much better substrate for dephosphorylation by PP1. So high glucose 6-phosphate essentially signals plenty of building blocks available, helping to push synthase towards its active unphosphorylated A form.
This PP1 enzyme seems to be cropping up everywhere. It takes phosphates off phosphorylase to inactivate it and off synthase to activate it. It sounds like a central hub.
It absolutely is. PP1 is a pivotal player, a central coordinator. It removes phosphate groups from phosphorylase kinase, from glycogen phosphorylase itself, and from glycogen synthase.
How does it manage all that?
Its activity is carefully controlled and targeted. The catalytic subunit of PP1 doesn't just float around. It's usually tethered to specific glycogen-targeting proteins. These proteins act like scaffolds, bringing PP1 together with its substrates right at the glycogen granule. There are different targeting proteins in muscle, like GM, and liver, GL, helping tailor the regulation to the tissue's specific needs.
And is there any crosstalk between the breakdown and synthesis pathways via PP1?
Yes, and this is really elegant. Remember how the active phosphorylated form of glycogen phosphorylase A is the signal for breakdown? Well, active phosphorylase A acts as a direct inhibitor of PP1.
Wow. So when breakdown is switched on high, it automatically helps switch off the enzyme that would activate synthesis.
Precisely. It ensures that glycogen breakdown and synthesis pathways are reciprocally regulated. They don't fight each other. When phosphorylase is active, PP1 is kept in check, preventing it from dephosphorylating and activating glycogen synthase. Only when phosphorylase activity decreases, for example, when glucose levels rise in the liver, does PP1 become fully active to promote synthesis. It's beautiful coordination.
That is really elegant. Okay, this brings us to the sort of global coordination picture, how the liver and muscle use these signals differently, but in an integrated way to manage glucose for the whole body. Let's take the well-fed state first, after a meal.
Okay, so after a carbohydrate-rich meal, your blood glucose rises. This triggers the release of insulin from the pancreas.
Right.
In the liver, insulin signaling dominates. It leads to the inactivation of GSK3 and the activation of PP1. This results in the dephosphorylation and full activation of glycogen synthase. At the same time, the high glucose entering the liver via the GLUT2 transporter binds to phosphorylase A, causing its inactivation by PP1.
So synthesis on, breakdown off.
Exactly. Glucose pours into the liver, gets phosphorylated, and is efficiently channeled into glycogen storage, helping to bring high blood glucose levels back down towards normal.
And in muscle, what does insulin do there?
In muscle, insulin has a major effect of triggering the movement of glucose transporters, specifically GLUT4, from inside the cell up to the plasma membrane,
Increasing glucose uptake.
dramatically increasing glucose uptake from the blood. Insulin also activates PP1 in muscle, leading to glycogen synthase activation. So muscle takes up glucose and stores it as glycogen for its own future use, also helping to lower systemic blood glucose.
Okay, now the opposite scenario. Fasting. Low blood glucose.
During fasting, blood glucose drops, and the pancreas releases glucagon. Importantly, muscle cells don't have glucagon receptors, so this signal primarily affects the liver.
Ah, okay. Liver only for glucagon.
Right. In the liver, glucagon triggers that TMP cascade we talked about, activating pKa. pKa phosphorylates and activates phosphorylase kinase, leading to activation of glycogen phosphorylase breakdown on. Simultaneously, pKa phosphorylates glycogen synthesis off.
Maximizing glucose release.
Exactly. Glucagon signaling also promotes gluconeogenesis, making new glucose, and inhibits glycolysis in the liver. The net effect is a strong push towards releasing glucose from the liver into the blood to maintain levels for the brain and other tissues.
And the final scenario. Fight or flight, triggered by epinephrine.
Epinephrine affects both liver and muscle. In the liver, it acts similarly to glucagon, promoting glucose release into the blood. But in muscle, which lacks glucagon receptors, epinephrine is the main hormonal signal for glycogen breakdown.
To fuel muscle activity.
Yes. Epinephrine activates the pKa cascade in muscle, turning on glycogen phosphorylase and turning off glycogen synthase. It also promotes glycolysis, generating ATP rapidly for muscle contraction. Remember, calcium released during contraction also directly boosts phosphorylase kinase activity. Muscle uses its stored glycogen selfishly and rapidly for its own high-energy demands during stress or intense activity.
It really paints a picture of different tissues responding to the same or different signals based on their specific roles. So what does this all mean when we step back and look at the bigger picture of metabolic integration?
It means that these pathways, like glycogen metabolism, while seemingly focused on one molecule, are deeply interwoven with overall energy balance. They are overlaid with these incredibly precise, often redundant regulatory controls, hormonal, allosteric, phosphorylation cascades that are exquisitely sensitive to the body's changing metabolic circumstances.
Adjusting the flow constantly.
Constantly adjusting the flow of metabolites, often without even causing huge changes in the concentrations of shared intermediates. It's all about maintaining stability or homeostasis while still being incredibly responsive. This whole system integrates seamlessly with the metabolism of other fuels, like fats and amino acids, ensuring the overall energy needs of the entire organism are met efficiently. It's a beautifully integrated system.
We've certainly covered a lot of ground today. We've navigated the remarkable world of glycogen metabolism from its unique, highly branched structure that enables that rapid energy access.
Yeah, those non-reducing ends.
To the sophisticated, multi-layered interplay of enzymes, hormones, and those allosteric effectors that ensure your body always has the glucose it needs, exactly when and where it needs it.
It really is remarkable, and maybe here's a final thought to consider. A system as seemingly straightforward as just storing glucose has evolved these incredibly intricate, almost redundant layers of control. You have cascades, allostery, compartmentalization, hormonal signals.
Right, so much complexity.
So much complexity. What does this profound level of control tell us about the fundamental importance, the absolute criticality of maintaining glucose homeostasis for the survival of vertebrate animals like us? And maybe how might even slight imperfections or miscommunications in these finely tuned regulatory pathways lead to significant metabolic disorders like diabetes or the glycogen storage diseases?
That's a great point. The complexity underscores the importance. Thank you for joining us on this deep dive today. Keep asking those why questions. Keep exploring the incredible molecular machinery that keeps us all going.